Well, thank you, Archer. It's a pleasure to be here today to share my story as told in To Mars with Love. So, you'd like to know if there's life on Mars? Well, many have wondered about that since Mars was first discovered thousands of years ago. First slide, please. No one is credited with discovering Mars, although Galileo was the first to observe Mars with a telescope in 1610. Mars is about half the size of Earth and around 140 million miles away on average. Due to its red appearance caused by iron oxides, it was named after the Roman god of war. Next slide, please. In 1895, Percival Lowell wrote a book titled Mars, in which he described artificially created canals on Mars. I actually remember hearing about those canals in my grade school years in the 1940s. Lowell later wrote a book in 1905 called Mars, the Abode of Life, where he presented fascinating speculations that Martian life forms may be even more highly evolved than those on Earth. Well, perhaps those speculations were inspired by H.G. Wells' 1898 novel titled War of the Worlds, in which he fantasized Martians invading Earth. On October 30th, 1938, Orson Welles aired War of the Worlds as a news story, periodically interrupting his regular radio program with an update of the invasion. Apparently, it was very realistic because the next day, Halloween, the front page story of the New York Times described waves of hysteria resulting from the newscast, where some people actually fled the area in their cars. But not until 1970 did Congress appropriate funds to actually go to Mars to look for life. These funds would be for the 1976 Viking Mission, next slide please, which would be the very first spacecraft to land on the surface of Mars. It was funded as a biological mission to determine whether or not there really was life on the red planet. And as of today, it is the only mission to have directly searched for extant life on Mars. Actually, I'm amazed that it was funded as a biological mission because almost nothing was known about Mars in 1970. There had been flybys in the 1960s by Mariners 4, 6, and 7 that showed a myriad of craters along with polar ice caps consisting of frozen carbon dioxide, or dry ice, but there was absolutely nothing suggestive of life from these flybys. Anyway, to address the life issue, NASA selected several experiments, next slide please, to search for the microbial metabolism on the surface of Mars. These experiments would be contained within 1 cubic foot, weigh not more than 22 pounds. And for those of you not born at the time, probably a good many of you in the audience, that was quite a requirement. Nothing like this had ever been attempted before. There weren't any fax machines in 1970. There weren't any PCs in 1970, and there weren't even any fax machines. They hadn't been invented yet. The Viking missions, in fact, marked the onset of technological miniaturization and the onset of the exciting new field of astrobiology. Well, one of the experiments selected was the label release life detection experiment, the LR, which was originally designed by Gilbert Levin. At the time, I was an assistant professor at Hopkins with expertise in biochemistry, biophysics, and the use of radioactive isotopes as tracers, and I was looking to make a career move. Well, when Gil Levin offered me a job in 1970 looking for life on Mars, it really blew my mind. It was so far out. My colleagues thought it was a suicidal career move, and I didn't disagree. But it seemed like fun and a perfect fit for my expertise and an exciting opportunity. So, confident that I could land on my feet if it was a total career disaster, I left an academic career behind and headed for Mars, becoming Gil's co-experimenter as I came up to speed. Well, the results of this experiment on Mars, which were actually positive for microbial life, have been published many, many times. But the behind-the-scenes story of how this experiment got there has only recently been published in my book, To Mars with Love. Next slide, please. As the name implies, it's meant to be a fun book and includes not only the science and engineering of the LR experiment, but all the humorous moments, photographs of the key players, mission memorabilia, and my parallel adventures in the equestrian world and beach communities of California. And the interactions between those communities and the Viking mission was sometimes quite amusing. Well, prior to the mission, I worked hand in hand with the engineers building the biology instrument, and my book documents all the challenges, crises, successes and failures, and the humor involved in getting the LR to Mars. And at the end of my talk, I will tell you how to obtain a signed copy of my book. But before getting into how the instrument worked, let's start at the beginning by telling you about the labeled release experiment itself. The concept of the labeled release experiment is very simple. Next slide, please. The experiment seeks heterotrophic microorganisms in soil. Now, don't let that scare you. What that means is that they were looking for microorganisms with animal-like metabolism, where organic substrates are consumed and the end product of metabolism is carbon dioxide. We all consume food and exhale carbon dioxide. A drop of liquid nutrient containing organic substrates is added to a soil sample, and if there are microorganisms in the soil, they metabolize the organic substrates and evolve carbon dioxide. Now, if those organic substrates are labeled with radioactive carbon, the evolved carbon dioxide will also be radioactive. So yeah, all you have to do to detect metabolic activity is put some sort of a Geiger counter over the headspace above the soil sample and monitor for the evolution of radioactive gas. Now, this is a typical active response under terrestrial conditions, and you can see it's a very rapid, strong response. When you run out of organic substrates, you will see a plateau. Now, to show that this active response is actually biological, a duplicate sample of the same soil is heat sterilized in advance of the nutrient injection. Failure of the controlled sterilized sample, which is down here, to evolve radioactive gas indicates that the heat treatment killed the soil microorganisms and that the active response is biological. The method was versatile in detecting microbes in a wide variety of soils, and it was extremely sensitive. Well, once the LR was selected to fly, the experimental conditions had to be adapted to a flight instrument yet to be designed. Next slide, please. Early tasks were to define the most effective heat sterilization temperature for that control. It had to be hot enough to kill the microorganisms, not hot enough to harm the hardware. After extensive testing with several soils, we finally settled on a control regime of 160 degrees centigrade for 3 hours. We also had to select the organic substrates appropriate for the flight experiment, and I'll come back to that in a minute. Once the nutrient was designed, we used it to develop a library of responses from soils from all over the world. Martin Marietta had the overall responsibility for the Viking lander, but the biology instrument was subcontracted to TRW in Redondo Beach, California. And as the design evolved there, we interacted with TRW to ensure that the instrument they designed actually did perform the LR experiment. Software also had to be designed. ROM sequences were developed by TRW to automatically implement the experiment on Mars. In addition, I worked with personnel at Martin Marietta to design computer programs for handling and manipulating LR data once it was returned to Earth. Remember, this was in the 1970s. There weren't any PCs. At the time, I had to plot all graphs by hand and calculate slopes by hand. So having a computer to do that was a very exciting improvement. Next slide, please. Selecting the organic substrates took about a year. There were 2 amino acids, glycine and alanine. There was also formate, the carbohydrate lactate, and glycolic acid, all universally utilized in a wide variety of soils. All these substrates had to be stable to heat sterilization and long-term storage. That was important because the nutrient had to be prepared a few years in advance of when it would be used on Mars, and it had to undergo several heat sterilizations before it ever left Earth, including sterilization of the entire spacecraft just prior to launch. In fact, this is the only spacecraft to have been sterilized. We didn't want to go to Mars to detect terrestrial hitchhikers. But the neat trick was that every carbon in each substrate was labeled with the same amount of radioactivity. That meant activity would be detected regardless of which carbon it had been derived from, and that the amount of gas released could be quantitated. Well, when we performed the LR experiments in the lab, we used a technique that allowed us to perform maybe 30 simultaneous runs in a day, which allowed us to compare many experimental conditions in a relatively short period of time. Well, performing the experiment in the laboratory is one thing. Getting it to work autonomously in a hostile environment millions of miles away is anything but trivial. We learned just how hostile the environment was when Mariner 9 went into orbit around Mars in November 1971, and I was on that flight team also. The Mariner 9 was the first spacecraft ever to orbit another planet. Next slide, please. We found that Mars was cold with temperatures rarely above freezing, with daily temperature fluctuations of more than 100 degrees centigrade. The surface was bombarded with solar radiation, and the atmosphere was almost entirely carbon dioxide. Atmospheric pressure was near vacuum, 6 millibars as opposed to 1,000 millibars here on Earth. And there was no liquid water found, but plenty of photographic evidence that liquid water had existed once upon a time in ancient times. Here is a Mariner 9 image showing dendritic patterns, which gave us hope that life may have evolved in ancient times when conditions were more favorable, and if so, might still be there. So how did the labeled release flight instrument work? Next slide, please. The next slide shows a simplified schematic of the LR hardware. Here we have a test cell, which can be raised or lowered from its cover, or head end as it was called. Once nutrient was added to the soil, any gas evolved passed through a 13-inch tube to a detector chamber containing 2 solid-state beta detectors that would monitor evolved radioactivity. Any carbon-based gas would have been detected, whether it was carbon dioxide, carbon monoxide, methane, or whatever. The tube was bent in several paces to prevent contamination of the detector by any radioactive particles escaping from the test cell. And both the head end and the detector chamber contained heaters. The head end heater would maintain the test cell between 10 and 12 degrees centigrade during an experiment, and could also be used to sterilize the soil at the 160 degrees centigrade for the control experiments. The experimental sequences on Mars would be performed in the dark under Mars atmosphere with an overpressure of helium. That was necessary to keep the liquid nutrient from exploding when added to the test cell, because Mars atmospheric pressure was near vacuum. Next slide, please. And here I am in 1971 with the very first rendition of the LR instrument, known as the Phase 1 breadboard. As you can see, here is the test cell and the tube going up to the detector. And here is my right hand is handling a lever that raises and lowers the test cell from the head end. The lever wasn't flight-like, but the test cell tube and detectors were. We used this early instrument to test for leakage and counting efficiency, and to run some soil tests under terrestrial conditions. It was slow going because each run took days and there was only one of these prototypes available. One of the first problems we ran into was that following a run, residual radioactivity adhered to the stainless steel head end and detectors, and the only way we could find to clean it up was by taking it apart and swabbing it down. But that wouldn't work on Mars. So after a lot of trial and error, we learned to clean it up by purging with helium while activating the detector and the head end heaters. Eventually, we got results that closely resembled soil tests done by our laboratory technique. Next slide, please. We tested 3 standard soils, Aiken, Death Valley, and Creek Bed. The solid lines are active responses and the dotted lines are sterile control responses. The graph on the left shows results from our standard laboratory technique, and the right shows results from the phase 1 breadboard. And as you can see, there's a pretty good match, which gave us confidence that the instrument as designed could adequately perform an LR test. Next slide, please. Well, the next rendition was the LR Test Standards Module, or the TSM as we called it, which was ready for use at TRW by December of 1972. This version contained all the essential components of the flight instrument with the test cell and the detector in this area. And once soil, and it's enlarged over on this side, once soil was placed in the test cell, a bell jar was placed over the entire thing and evacuated to Mars atmospheric conditions, which took about a day. Then it took a day or two for a soil run and another day for cleanup. All operations were done manually by flipping switches on the instrument panel. It was slow going and we kept this instrument running 24/7. If something needed doing in the middle of the night, either I or my full-time technician had to be there to do it. And it was a really touchy instrument requiring much attention, upkeep, and repair. Next slide, please. Well, the way the instrument initially worked is shown in this slide. There are 4 chambers, a test cell, detectors, a water reservoir, and a nutrient reservoir. And there's a high pressure helium source and a low pressure helium source to push things around. There were a series of 8 valves, 1, 2, 3, 4, 5, 6, 7, 8. And they were opened or closed on 1-minute intervals or multiples of 1 minute. Initially, valve 46 over here was open to break a glass ampule in the nutrient reservoir. And then low pressure helium was bubbled through the system to remove any products of radioactive disintegration that had accumulated since preparation of the nutrient. Before nutrient injection, this valve 61 was opened for 1 minute to allow equilibration of the test cell headspace and the water vapor pressure in the water reservoir. The purpose was to raise the atmospheric pressure in the test cell and prevent the liquid nutrient from exploding when added to the test cell under Mars atmospheric conditions, which again are near vacuum. This turned out to be a total disaster and eventually this whole system was removed, a story detailed in my book. And instead, we pressurized the cell with low pressure helium. But although this system was physically removed from the hardware, valve 61 remained in the ROMs, and it became known as the phantom valve. Well, to deliver nutrient to the soil, valves 46, 59, and 45 were opened to high pressure, so high pressure helium could drive the nutrient through a 3 mil restrictor right here to the soil during the 1 minute time the valves were open. All valves were then closed and the experiment begun. Note that this was a timed delivery and the amount of nutrient delivered was determined by sizing that restrictor. Following an experiment, radioactive gas in the test cell was removed by purging with low pressure helium through valves 42 and then out 52. To clean up any residual radioactivity on the head end, purging with low pressure helium continued while the heaters in the head end and the detector chamber were turned on. Well, I went to TRW for a couple of weeks in December 1972 to learn to use the TSM and to make some soil runs. Oh, the results were so disappointing. They didn't match results obtained in our laboratory technique, and we couldn't even get the same result twice in a row. We had to revamp our plans and devote full time to solving engineering problems. And we had to do it without changing the basic instrument design. We were told that it's too late for hardware design changes. Well, the only option we had to solve problems was by changing experimental sequences and procedures. And over the next few months, we made more than 20 sequence and procedural modifications. These are many of which are documented in my book. And by April 1973, the LR experiment in the TSM was working pretty well under Martian conditions. Next slide, please. Meanwhile, the flight instruments were being built at TRW. The flight instrument is much smaller than the TSM, with all 3 life detection experiments contained in 1 cubic foot, each with its own module. Unlike the TSM, which was designed for repeated use, the flight instrument was a one-time use instrument. The LR module is here in the center, and the other 2 life detection tests on each side of it. There was a 4th module, which you can't see here, called the Common Services module, which contained things used by all 3 experiments, such as the high and low pressure helium sources. And here in the center is the soil distribution assembly, which distributed soil to each experiment. The LR module contained 1 head end and 4 test cells on a carousel. Any specific test cell could be rotated under the soil distribution box to receive soil and then rotated back under the head end, where it would be raised and sealed against the head end for an experimental procedure. And over here on the right is an actual flight instrument with its 4 modules. It was assembled under sterile conditions. Again, we did not want to go to Mars to detect terrestrial hitchhikers. TRW built 6 of these instruments. 3 were used for pre-mission testing, 2 went to Mars, and a 3rd was a spare. The next slide shows where the biology instrument was positioned on the lander. Here is the soil processor with the biology experiments directly under it, but they were out of sight. And here is the sampling arm that went out and grabbed the soil. And here are the 2 cameras. And here I am next to the spare lander that was set up at the Jet Propulsion Lab during the mission. Well, by November of 1973, TRW was ready for the first full test of all 3 life detection experiments in the very first flight instrument. The instrument was placed in a chamber about 10 cubic feet, which was then evacuated to Mars atmospheric conditions, which took days. Next slide, please. All 3 life detection tests failed. That's when the fun really began. NASA personnel and Martin Marietta personnel descended on TRW and took over management of the biology instrument, and they loved that. I clearly remember Jim Martin, NASA's manager of the entire Viking mission, slamming his fist on the table and saying, "We'll throw biology off the mission." Well, somebody reminded him that Congress had funded Viking as a biological mission and he just couldn't do that. So he slammed his fist on the table again and said, "Okay, we'll throw off the worst 2 experiments." Well, the problem was nobody could figure out which were the worst 2. Gil and I got letters saying that all key scientists will remain in residence until the problems were solved. Gil had a company to run, so that meant me and my technician, of course. I had gone out there in November of 1973 for a 6-week trip and never got home until spring of 1975. The two major problems for the LR were that the nutrient injection system failed because that 3 mil restrictor clogged, and the sterilization temperature for the control sequence only reached 140 instead of the required 160 degrees. And those were only the major problems. There were many other less critical ones. Solutions had to be within the constraints of the existing hardware and software. It was too late to redesign. So solving problems meant creative thinking outside of the box about new procedures and sequences, and then testing those candidate changes in the TSM. Well, everybody worked like crazy. There were nonstop crisis meetings starting at 7 a.m., 7 days a week to figure out what to do. We kept the TSM running 24/7, testing candidate fixes. We even pulled out the phase 1 breadboard for some of these tests, which was real desperation. For the nutrient injection system, next slide please, it took months, but we finally managed to fix it within the constraints of the existing system. We did it by removing the restrictor and adding a valve to the nutrient delivery system, which changed it from a time delivery to a volume delivery using a metered cavity. Well, bet you're wondering how we did that without changing the hardware design. Remember the phantom valve? Well, because it was in the ROMs, we were able to add it back into the system and create a metered cavity that delivered the correct amount of nutrient. How we manipulated that phantom valve to solve the problem is a fascinating story detailed in my book. The other major problem was that the sterilization temperature was too low. No other heater could be found that would reach the required 160 degrees, but the problem was eventually solved by adding a booster heater to the head end that would raise the sterilization temperature from 140 to 160. And this became important during the mission. Well, by January of 1975, a year and 2 months after those failed tests, we were finally ready to test the life detection test experiments in the next available flight instrument using all the new sequences and procedures worked out in the TSM. Unfortunately, the LR test in the second flight instrument was another dismal failure because the nutrient ampule failed to break. All other systems worked fine, but the LR was a big disappointment. Well, we had one last chance to show that the LR actually worked in a flight instrument. A third flight instrument is available for testing in the spring of 1975, just a few months before launch. Amazingly, in the third flight instrument, the LR worked. Next slide, please. Here we see the results with Aiken soil, one of our standard test soils, tested under Martian conditions. We had a typical positive response following first injection and an additional response following a second injection of nutrient. These dips in radioactivity reflect turning off one or the other of the two beta detectors to test single channel counting modes. We also had a typical control response down here in which the soil was first sterilized in the test cell at 160 degrees for 3 hours. It was close. The final parts of this test were completed as the two final flight instruments were being shipped to Cape Kennedy for incorporation into the spacecraft. Well, the first Viking spacecraft was launched on August 20th, 1975. Next slide, please. And it arrived at Mars on June 19th, 1976. Everyone gathered at JPL in June, where an entire building had been dedicated to Viking. On July 20th, 1976, the lander was released from the orbiter sometime around midnight. A parachute was deployed around 21,000 feet above the surface, and retrojets were fired about 50 feet above the surface to slow it down. The entire biology team stayed up all night waiting for the landing. Next slide, please. Here's Gil Levin here and here, and Chuck Klein, who was the biology team leader, and all the other members of the team, which are, they're all labeled in my book. But as the lander approached the surface, we watched numbers flashing on a monitor, giving the altitude of the lander above the surface: 1,300 feet, 650 feet, 50 feet, silence. I was sure it was coming in too fast and had crashed. But then we have touchdown, came over the loudspeaker. Within a minute, the very first image from Mars, next slide please, flashed on the monitor. Well, we couldn't make much sense out of this picture, and the image then built up slowly to the right, a few sections at a time, and finally the lander footpad appeared. Next slide please. And everyone realized that we actually were looking at the surface of Mars. This is the very first picture that ever came back from Mars. Champagne bottles magically appeared and everyone was in very high spirits. Well, the first color picture came in a few hours later. Next slide, please. It showed a barren landscape not unlike terrestrial deserts, and the red color reflects iron oxides. There are no obvious life forms, no camels, although there certainly could be microorganisms in the soil. There were tantalizing greenish patches on some of the rocks that generated some heated discussion, but the imaging team eliminated the problem by adjusting the colors so they no longer appeared green. That was not the end of the story, however. Well, the first soil sample was collected on Sol 8. Sol is the Martian day, and the LR got underway on Sol 10 when nutrient was first injected onto soil. The first 9 hours of data were available around 7:30 that evening. I sat down at the computer surrounded by Gil and other members of the biology team and put in the commands for a printout of radioactivity evolved over the next few hours. Next slide, please. Lo and behold, it was clearly a positive response. Equally amazing, the instrument actually worked. That was a bit of a surprise. It was a really exciting time, and everyone present signed the printout. Well, that first cycle ran for 7 sols, after which the critical control was run, in which a duplicate sample of the same soil was heat sterilized at 160 degrees C for 3 hours before nutrient was added. Next slide, please. Another big surprise, the response of the control was negative. And taken together, the results from these 2 cycles on Lander 1 were consistent with a life response and looked a lot like a terrestrial life response. Next slide, please. And here you can compare that the Mars results from the first 2 cycles with the slide I showed you earlier of a terrestrial soil under terrestrial conditions. The magnitude of the response was much, much lower than this soil, but that was not unlike responses we had seen from low population soils, such as those that we had collected from Antarctica. Well, everybody immediately jumped on the bandwagon of trying to explain the results by some exotic chemical reaction. Nobody, including me, was willing to accept the life interpretation as conclusive, mainly because nothing else on the mission indicated life. The other 2 life detection tests were negative. The organic analysis experiment failed to find organics, and every theory of life said that where there's life, there has to be organics. And there was no liquid water found on Mars, and water is believed essential for life. For the remainder of our runs on Landers 1 and 2, we tried to learn more about this active agent and did everything we could think of to figure out whether the active response was biological or chemical. I'm not going into detail about those results, but the next slide summarizes all the results. The active agent gave a positive response when injected with carbon-14 labeled organic substrates. The same active response was obtained by the second lander 4,000 miles away. So the active agent, whatever it was widespread. The active response was also repeated at both landers. The activation was not caused by ultraviolet exposure because we took one of those samples from under a rock and it was fully active. Now for the thermal controls, it was inactivated by preheating at 160 degrees for 3 hours, which you've already seen. And it was partially inactivated by preheating around 50 degrees centigrade for 3 hours. In trying to distinguish whether the active result was biological or chemical, we came up with a bright idea. We figured out a way to reduce the sterilization temperature from 160 to 50 degrees, something that had never been tried on the TSM or even thought of prior to the mission. So how did we do it? Well, the story's in my book. The idea was that very few chemicals are sensitive to 50 degrees, which is 122 Fahrenheit, but Martian microbes adjusted to much colder temperatures might be adversely affected. 2 runs were conducted on Lander 2 by pre-treating the soil around 50 degrees for 3 hours. Both results show that pre-treatment around 50 degrees dramatically reduced the magnitude of the response by about 70% or more. This was strong evidence for biology. The active agent was also inactivated by long-term storage at approximately 10 degrees C. For our last runs on each lander, we used soil stored in the hopper at 10 degrees for around 84 days on one lander and 140 days on the other. There was essentially no activity in either one, showing that the active agent is destroyed by long-term storage around 10 degrees C. This also strongly favors biology for the same reason. Namely, few chemicals are affected by 10 degrees, which is 50 degrees Fahrenheit, but biology on Mars very well might be. So, did the label release find life on Mars? Next slide, please. It's still a highly controversial question, even after 40 plus years. The results are entirely consistent with biology, especially the thermal controls where 50 degrees partially destroyed the positive response and 10 degrees essentially destroyed it. Very few chemicals are destroyed by such low temperatures. The problem is that nothing else on Viking was positive for biology. The other life detection experiments were negative, no organics were found, and no liquid water was found at that time. Some scientists, including myself and Gil Levin, the principal experimenter, believe the evidence strongly favors biology, but others disagree, attributing the results to some as yet unknown exotic chemical. Bear in mind that many have tried over 40 plus years to replicate the labeled release results non-biologically. None have succeeded. So the LR results have remained controversial even after 44 years. Recent missions support the biological interpretation. Next slide, please. Water has now been found. Not much, but perhaps enough to support life. Complex organics have been found. Methane has been found, which is a possible indicator of life. Why? Well, because the half-life of methane in the atmosphere is only around 20 some odd years, so its mere presence indicates that it must be constantly replaced. Almost all the methane in the Earth's atmosphere results from the action of methane bacteria. And most interesting about the methane on Mars is that it is localized and its concentration cycles with the seasons, a mystery entirely consistent with biology. Well, elevated oxygen levels have just recently been reported, again, localized and cyclical with seasons. Keep in mind that there could be life on Mars, whether or not the LR detected it. Nothing about the Mars habitat precludes life. Terrestrial extremophiles exist that could possibly exist on Mars? And if so, that life is microbial. There's no macroforms that have been observed, no little green men up there. But I leave it to you to read about it and decide for yourself. The evidence pro and con is presented toward the end of my book. If the LR really did detect life, that would perhaps be the most astonishing finding ever. If not, the LR story is still an amazing account of an incredibly ambitious, first ever and only effort to put a metabolic experiment on another planet, an achievement that has not since even been attempted. So maybe there really is extant life up there, whether or not the LR found it. It has been so disappointing to me that no further life detection experiments have been sent to follow up on the exciting LR results. All missions since Viking have looked for water and possible habitat. None have directly sought life. Hopefully, future missions will directly look for life, and when they do, Mars may yet have big surprises. What I have learned from Mars and from discoveries that I read elsewhere in the universe is, next slide please, expect the unexpected. And that statement applies not only to future space missions, but also to the tales and situations presented in To Mars with Love. Thank you. I would be happy to answer questions, but before I do, I'd like to offer you a signed copy of my book at a special discounted price that includes free shipping within the United States. All you have to do is send me an email at info@tomarswithlove.com, mention Balticon in the subject line to get this discount, include your address and any special requests you may have for inscriptions, and I will send you a custom PayPal invoice that reflects the discount and the free shipping. The offer is good until March 15th. And we could stop the recording now, and I'll be happy to answer questions. Thank you.